looked at passive transport, and passive transport simply means is a kind of transportation that does not require energy. And the reason it does not require energy is because the molecule is transported from higher concentration to lower concentration. Now, there are some points that some molecules are still transported from higher concentration to lower concentration, but because the molecules cannot pass through the cell membrane by itself, it will need the help of a carrier. So like now, when you want to transport glucose, because glucose cannot pass through the cell membrane by itself, it will need a carrier, even if it is moving from high to low. Sometimes you want to transport calcium ion or potassium ion because they have charges. They cannot pass through the cell membrane by themselves, so they need a carrier. So we call that kind of transportation, we call that kind of transportation facilitated diffusion or facilitated diffusion. Is a kind of diffusion that does not require energy, but it requires a protein. So this require protein, but it does not require energy. All right. So you want to keep this somewhere it is no energy is required. Protein is required, but no energy is required. So if you look at this, for instance, now, this molecule is transported from higher population. This is higher concentration here, and this is lower here. So it's moving from where there is more to where there is less. So it doesn't require energy. So looking at this as well, here you have more of these molecules here, and it's being transported to where it is low. So it is not going to require energy. However, it requires a transporter. All right. Now, channel protein. So a channel protein provides corridors. Like I told you earlier, it is channel. So like aquaporin, for instance, is a kind of channel protein. Some ion channel protein also exists. We have sodium ion channel protein, potassium ion channel protein, and they help to move potassium or move ion. This helps to move water. This is aquaporin. Just showing you an example. All right, so it's moving from high to low. It still does not require energy. It is a diffusion. What we call it facilitated diffusion. All right, now there are some gated channel. Some ion channels are called gated channel. They are called gated channel because they are going to open and they close. So there's sodium ion gated channel. Those gated channel opens and close. So for example, due to stimulus, for example, in nerve cells, potassium ion channel open in response to electrical stimulus that's coming from the brain. Other gated channel open in response to chemical stimulus binding of a specific substance to the protein. So we call those carrier protein. This is a carrier protein. It open, changes shape, and then deliver the substance that is carrying. All right. Now we have the other kind of proteins, transportation that we call active transport. Again, I've talked about passive transport, diffusion, osmosis, and then facilitated diffusion. Facilitated diffusion does not require energy, so it is still passive. It only requires a protein. Now, when do we talk about passive active? We only begin, again, this is passive because it's going from high to low. All right. Now we begin to talk of passive whenever something is coming from where it is low to where it is high. That would be passive. That would be active because it requires energy because it's now going against concentration gradients. Now look at this, for instance. Now, this is an example of active transport. And see, when you are transporting, you see this cube here, think about this as sugar. When you are transporting this sugar from here into the cell, now where do we have more sugar, more of the cube? We have more of the cube right inside the cell. There are just 2 of them here, whereas there are 5 here. So what is logical, there will be passive transport, is if it is moving from inside the cell to outside the cell, that will be passive transport. But now because it's moving from where it is low to where it is high, it is called active transport. It's going to require energy. And so you see energy waiting here. Energy is going to be required for this process to occur. So active transport requires energy. Passive transport does not require energy. And it usually requires energy in the form of ATP hydrolysis to move substances against their concentration gradient. You see that? Against their concentration gradient means it is moving from where it is low to where it is high. All proteins involved in active transport are called carrier proteins, just like you can see here. Now, sodium-potassium pump also does active transport. Because this is it. The rule is this: whatsoever it is, anytime 3 sodium, anytime that you have 3 sodium, I think I will show you on this image. So anytime you see 3 sodium goes out of the cell, you are going to see 2 potassium come in. Regardless of the quantity of potassium or sodium, as long as sodium is pumped out of the cell, 2 potassium must come in. It's a rule. If 3 sodium goes out, 3 potassium must come in, regardless of the quantity of potassium in the cell. If there are just 2 potassium there, they must come into the cell. That kind of transportation is active transport. All right? It is also called active transport because it requires energy. Now, this is passive, just a way of showing you a review. Passive, diffusion, no energy is required. Osmosis, no energy is required. Facilitated diffusion, no energy is not required, but protein is required. In this case, active diffusion, active transport is active because it requires energy, because it's moving from low concentration to high concentration. All right? When we look at membrane potential, we are talking about the charges, the voltage on the membrane, and how it changes over time, and what is responsible for the gradient or the differences in the voltage on the membrane. That is what we call membrane potential. Electrogenic pump helps to transport protein that generate voltage across a membrane. It helps in storing energy that can be used for cellular work. Now, the main electrogenic pump differs between plant and animal. In animals, we have the sodium-potassium pump. That is an electrogenic pump. It helps to circulate or to modulate the charges, the polarity, or the charges on the membrane. In plants, fungi, and bacteria, we have what we call the proton pump, all right, instead of the sodium-potassium pump. Now, co-transport is when an active transport of a solute indirectly drives transport of other substances. Now, this is called co-transport, and it's simple. It's more like when sodium is moved in now, it generates a power, and the fact that sodium is moved in, then it's allow glucose to be moved in. Just think about it like this. Let's say a big truck is driving very fast. Now, if a car is just, let's say you have this very small car just parking, and a big truck is driving at a very fast rate, what's going to happen? The pressure that the truck is driving is going to stylishly move the small car. That is, in simple terms, the force that truck is driving invariably attracts some force also to the car. That is kind of co-transport. When I want to joke with co-transport, I also use this, that if you are coming into my class and you are pregnant, that is co-transport, because the movement of one person has brought in the baby into the class. That is co-transport. So co-transport occurs when active transport of a solute indirectly drives the transport of other solutes. I'm going to show this to you. So this is what we call co-transport. So plants use proton pump to generate hydrogen-helium gradient across the cell membrane. A co-transport couples the movement of hydrogen-helium back down to its concentration gradient to the active transport of sucrose into the cell. This is how plant loads sucrose. So now, as this is moving, now you see this is diffusion of hydrogen. Now this movement invariably powers the movement of sucrose as well. This is called co-transport. When the movement of one substance generates energy to move the other substance, that is co-transport. I will talk about this now. You can please read this. Now, exocytosis. Exocytosis simply means exit. It's something is moving out of the cell. So you see, this is the product from Golgi apparatus binding to the cell membrane, and the product leaves the cell. That is exocytosis. Exocytosis simply means something is going out. The opposite of exocytosis is endocytosis. Endocytosis is when a substance is coming into the cell, and that have three types. Endocytosis are phagocytosis, pinocytosis, and receptor-mediated cytosis. Phagocytosis means there is food coming into the cell. Pinocytosis means there's a liquid coming into the cell. And receptor-mediated endocytosis means that whatever wants to come into the cell need a receptor before it can come in. It needs something to receive it. All right. It's just like if there is someone that wants to enter my class, my office now, I have to receive the person and bring the person in. That is a receptor-mediated endocytosis. The office is locked. You can only enter the office if I bring you in. That is receptor. I have to receive the person and bring the person in. Receptor-mediated endocytosis. Now, this is phagocytosis. You see, for this one, now look at this. I think I'll just use this. You see this pseudopodium get invaginated into the membrane. The membrane push it in. That is phagocytosis because it has to do with large molecules, solid molecule entering the cell. Then lysosome is going to bind to it and break it down so that the cell can enjoy the particle that was just into the cell. Pinocytosis is when it is liquid. That's the difference. It's just liquid and solid. Phagocytosis, solid. Pinocytosis, liquid. Now, receptor-mediated endocytosis, you see, this here are the receptor, the red thing are the receptor. Then the solutes are going to bind on the receptor, and that's it. Then they come in just like the way it is for phagocytosis. The only difference is that these require receptors. Okay, this is just to compare the three. All right. So I will stop there. I mean, that's all anyway. I'm going to stop there.